As we get older, a nearly universal symptom starts to emerge. Simple cuts, scrapes, and minor injuries take noticeably longer to heal. It is easy to assume this delay happens because older cells are tired, or because the body naturally winds down over time. But the real cause is far more active. An actual structural blockade exists within the tissue, a literal roadblock preventing your body's natural repair mechanisms from functioning. The skin's inability to bounce back is a mechanical failure caused by these biological roadblocks, rather than an unavoidable side effect of passing time. The biological culprits creating this blockade are called senescent cells. These are damaged cells that stop functioning, but completely refuse to die off. Instead of clearing out of the way, they sit perfectly stagnant. They act like biological debris trapped deep inside the skin layer. This tissue diagram shows the problem's scale. In aged skin, broken cells can make up 15% of the population. They cause active damage, secreting molecules that trigger inflammation and break down structural collagen. These secretions suppress healthy cells, trapping the environment in chronic distress. The presence of these cells forces the skin environment into a state of permanent stalemate, effectively paralyzing any hope of natural repair. A team of researchers at Boston University looked at this process and asked a straightforward question. What would happen to aged skin if this cellular debris was simply forcibly removed? To find out, they turned to senolytics, a new class of cell-destroying drugs designed specifically to clear out these biological roadblocks. The specific drug used in the trial was a repurposed cancer treatment called ABT-263. It has a highly unique ability. It hunts down only the broken senescent cells, while leaving healthy cells entirely untouched. The researchers applied ABT-263 directly to the aged skin of mice for a short, concentrated window of just 5 days. This short-term, highly targeted subtractive approach set the stage to see if the body could finally heal itself without interference. The immediate microscopic results showed that the markers for these stagnant cells in the mice plummeted, successfully clearing the biological blockade. Removing the broken cells reactivated dormant healing genes. They immediately began controlling clotting, growing blood vessels, and rebuilding collagen. The definitive proof: by day 24, treated mice achieved an 80% wound closure rate, compared to only 56% untreated. Removing the cellular debris provided a massive improvement. It biologically reverted the aged tissue back to a highly energized, youthful state of repair. There is a crucial scientific caveat to keep in mind. This research is based on early-stage mouse models, not finalized human therapy. This is partly because of a biological nuance. These stagnant cells actually perform a useful function during the very first stages of wound healing by sending out signals for help. The core challenge for future medicine is that treatments must be precisely timed. These cells should only be evicted when they overstay and become toxic. An ultimate cure will rely on the flawless, carefully timed eviction of these temporary workers, rather than total biological eradication. This discovery targets severe, real-world medical crises, extending the focus of senolytics into high-stakes clinical care. This subtractive approach could drastically accelerate healing times after major invasive surgeries, vastly improving recovery outcomes for elderly patients. It also highlights the potential to treat chronic, dangerous wounds that refuse to close, a critical issue specifically for diabetic patients, where current treatments frequently fail. Ultimately, medical recovery will rely on a subtractive approach, restoring our inherent healing capabilities by evicting the broken cells suppressing them.